In the crushing darkness, 2,000 feet beneath the ocean's surface, something impossible drifts through the void. At first glance, it appears to be nothing more than a ghostly thread, a translucent ribbon spiraling through the black water like a discarded fishing line caught in some invisible current. But as the submersible's lights wash over its length, the truth becomes horrifyingly clear. This is no debris, no accident of human carelessness lost to the depths. This is alive, and it stretches farther than the eye can follow, disappearing into darkness on both ends, a living rope that may extend for hundreds of feet through the silent abyss. This is the siphonophore, one of the ocean's most enigmatic hunters, and what you're witnessing is only a fraction of a single organism. Or is it? The answer to that question will challenge everything you thought you knew about what it means to be alive. The year was 2020, when marine scientists aboard the research vessel Falkor, exploring the Ningaloo Canyons off the coast of Western Australia, encountered something that stopped them in their tracks. Using their remotely operated vehicle named Sebastian, they descended into the twilight realm, where sunlight becomes memory and pressure begins to crush all but the most adapted life forms. What they found spiraling in the depths was a siphonophore of the genus Apolemia, forming a massive ring-like structure in what scientists described as a galaxy-like formation. The outer ring alone measured approximately 150 feet in length. The entire organism may have stretched nearly 400 feet, making it potentially the longest animal ever recorded by science. Longer than a blue whale, longer than any dinosaur that ever walked the earth. And yet, impossibly, its body was no thicker than a broomstick. When the researchers first shared footage of this discovery, marine biologists around the world reacted with a mixture of wonder and disbelief. Dr. Rebecca Helm, a biologist at the University of North Carolina, stated that in all her expeditions into the deep, she had never seen anything like this hunting formation. The creature wasn't simply drifting, it was hunting. The spiral structure wasn't random but deliberate, a floating death trap suspended in three-dimensional space, waiting for prey to swim into its embrace. But to understand what makes the siphonophore such a masterpiece of evolutionary design, we must first confront an uncomfortable question about its very nature. When you look at a siphonophore, are you looking at one animal or thousands? The answer exists in a gray area that biology struggles to categorize. A siphonophore is what scientists call a colonial organism, a concept so alien to our understanding of individuality that it borders on science fiction. Imagine if your heart, your lungs, your stomach, and your limbs were all separate creatures, genetically identical but physically distinct, connected by living tissue and utterly dependent on one another for survival. Now imagine that each of these specialized beings was conscious in its own microscopic way, performing its singular function in service of the collective. This is the reality of the siphonophore. The story of these creatures begins over 600 million years ago in the ancient Precambrian seas, long before fish evolved, before the first animals crawled onto land, before even the Cambrian explosion that would fill the oceans with complex life. The ancestors of siphonophores belonged to the phylum Cnidaria, the same ancient lineage that would eventually give rise to jellyfish, corals, and sea anemones. But somewhere in those primordial waters, something extraordinary happened. A single polyp, a simple tube-like organism with a mouth surrounded by tentacles, began a process of asexual reproduction through budding. This was not unusual. Many cnidarians reproduce this way, creating copies of themselves. But in the lineage that would become siphonophores, these copies refused to separate. They remained connected, forming chains, and over millions of years, something revolutionary occurred. These connected individuals began to specialize. What emerged from this evolutionary experiment was a form of biological organization so efficient, so perfectly adapted to the deep ocean's challenges that siphonophores have survived mass extinctions that wiped out countless other species. When the dinosaurs vanished 65 million years ago, siphonophores continued their silent drift through the abyss, unchanged and unaffected. When ice ages locked the planet's surface in frozen wasteland, the deep ocean remained constant and the siphonophores endured. They survived because they had discovered something profound about existence itself, that cooperation, taken to its absolute extreme, could transcend the limitations of individual life. The body plan of a siphonophore reads like a blueprint for an alien spacecraft rather than a living creature. At the front of the colony floats the pneumatophore, a gas-filled chamber that acts as both flotation device and compass, keeping the entire structure oriented properly in the water column. The gas inside isn't simply air, but a carefully regulated mixture that allows the colony to adjust its buoyancy with precision, rising or sinking through pressure zones that would crush most creatures. Behind the pneumatophore comes the nectosome, a segment studded with bell-shaped structures called nectophores that pulse in synchronized rhythm, driving the colony forward through the water using jet propulsion. These swimming bells work in perfect coordination, the smaller ones near the top handling fine adjustments and turning maneuvers, while larger ones at the base provide the raw thrust needed for maximum speed. Watch them pulse, and you're witnessing one of nature's earliest experiments with coordinated movement, a living engine designing millions of years before humans would invent the concept of teamwork. But the true horror and wonder of the siphonophore reveals itself in the siphosome, the long trailing stem that can stretch for hundreds of feet into the darkness. Along this stem, in repeating patterns unique to each species, hang the specialized zooids that make the siphonophore such a devastating predator. The gastrozooids are the feeding polyps, each one a living stomach with its own mouth, its own tentacle extending outward from the main body like fishing line deployed from an invisible trawler. These tentacles don't simply hang limp in the current, they are weapons. Every few inches along their length, side branches called tentilla extend outward, and on these branches hang millions of nematocysts, microscopic harpoons coiled under pressure, waiting to fire. The mechanism of a nematocyst is so violent, so explosive, that high-speed cameras capturing its deployment show events occurring in less than a millisecond. When prey brushes against the trigger on the surface of these cells, a tiny door bursts open and a coiled thread shoots out with acceleration forces exceeding 5 million Gs, faster than a bullet, puncturing the prey's body and injecting a cocktail of toxins. Some of these toxins paralyze the nervous system. Others begin breaking down cellular walls, pre-digesting the victim from the inside out. A siphonophore doesn't just kill its prey, it processes it while it's still struggling, still trying to escape tentacles that have already begun the work of consumption. Different species of siphonophores have evolved 4 distinct types of these nematocysts, each designed for specific prey and hunting conditions. Heteronemes are the largest, spear-like structures for piercing tough exoskeletons. Haplonemes are open-tipped tubules with spines, the most common type, acting as general-purpose weapons. Desmonemes lack spines entirely, but produce adhesive compounds that glue prey to the tentacle, preventing escape. Rhopalonemes have wide tubules designed to envelop and immobilize larger prey items. The siphonophore doesn't choose which weapon to deploy. It uses all of them simultaneously, a living minefield of different death mechanisms distributed across hundreds of feet of near-invisible tentacles.
But perhaps the most unsettling adaptation of deep-sea siphonophores is their mastery of light in a realm of absolute darkness. Many species are bioluminescent, capable of producing their own illumination through chemical reactions in specialized cells. The giant siphonophore, Preya dubia, attracts its prey with bright blue bioluminescence, a beacon in the black that lures small crustaceans and fish toward what they perceive as a meal, but is actually their executioner. Some species of the genus Irina have taken this deception even further, evolving red bioluminescent lures on their tentilla, organs that flick and twitch in patterns that mimic the movement of tiny copepods and other zooplankton. Red light has a long wavelength that many deep-sea creatures cannot see, making these lures invisible to potential predators but irresistibly attractive to prey who mistake the glowing movement for the bioluminescent signature of their own food. Scientists have documented fish swimming directly into these lures, mesmerized by what they think is an easy meal, only to be ensnared by tentacles they never saw coming.
The hunting strategy of most siphonophores is patience incarnate. In the deep ocean, where food is scarce and the distance between prey encounters can be measured in hours or days, energy conservation is survival. The siphonophore employs what biologists call a sit-and-wait strategy. The colony positions itself in the water column, adjusts its buoyancy to hover motionless, and deploys its tentacles in a deadly curtain or spiral formation. Then it waits, for hours, for days if necessary. Its gelatinous body requires minimal energy to maintain. It doesn't need to chase, it doesn't need to search. The ocean itself will deliver prey into its embrace. And when that prey makes contact with even a single tentacle, the response is instantaneous. The tentilla undergoes an explosive conformational change, wrapping around the victim like a spring-loaded trap, maximizing the surface area of contact for thousands of nematocysts to fire simultaneously. The prey never had a chance. By the time its nervous system registers pain, it's already been paralyzed and is being drawn upward toward the nearest gastrozoid for digestion.
What makes siphonophores even more remarkable is the evidence suggesting that different species have evolved to specialize in hunting different types of prey, and that they can shift between being specialists and generalists across evolutionary time. Research analyzing the morphology of tentacles from 45 different siphonophore species found strong evolutionary associations between tentacle structure and prey type. Species that hunt primarily copepods have evolved different tentacle and nematocyst configurations than those that specialize in catching fish.
And contrary to the long-held belief that specialization is an evolutionary dead end, siphonophores demonstrate that specialists can evolve into generalists, and that specialists on one prey type can directly evolve into specialists on entirely different prey, all by modifying the structures of their prey capture apparatus without changing their overall body plan. The deep ocean environment where most siphonophores thrive is one of the most hostile places on Earth. At depths of 2,000 to 3,000 feet, water pressure reaches over 90 times atmospheric pressure at sea level, enough to crush human bone like chalk. Temperatures hover just above freezing. No sunlight has penetrated these depths for millions of years. The water column is so vast, so empty, that the probability of encountering prey or predators in any given moment approaches zero. And yet, siphonophores not only survive here, but dominate as apex predators. Their secret lies in their gelatinous construction. Unlike fish with air bladders that would implode under extreme pressure, or mammals with lungs that would collapse, the siphonophore's body is mostly water, which is incompressible. Its gelatinous structure allows it to exist at depths that would destroy most complex organisms. But this adaptation comes with a terrible price. The siphonophore's body is held together by the very pressure that would kill other creatures. Their hydrostatic skeleton relies on external water pressure exceeding 460 bars to maintain structural integrity. This means that if you were to bring a giant siphonophore to the surface, it would quite literally explode, its body fragmenting into gelatinous pieces as the internal pressure that holds it together is suddenly removed. For centuries, when fishermen accidentally hauled up siphonophores in their nets, they found only shapeless blobs of transparent jelly, unidentifiable masses that gave no hint of the sophisticated colonial predators they had once been. It wasn't until 1887 when the Monterey Bay Aquarium Research Institute conducted the first systematic study of living Praia dubia in its natural habitat that scientists finally observed these creatures as they truly are, intact and hunting in the depths. The sensory capabilities of siphonophores remain one of the great mysteries of marine biology. They have no eyes, no centralized brain, no nervous system in the way we understand it, and yet they respond to their environment with precision that suggests some form of distributed awareness. The pneumatophore appears to function as a pressure sensor, allowing the colony to detect changes in depth and adjust its buoyancy accordingly. Chemical receptors distributed across the tentacles can detect the presence of prey in the water. But how does a creature with no central processing unit coordinate the firing of millions of nematocysts simultaneously? How do the nectophores synchronize their pulsing without a brain to conduct their rhythm? The answer seems to lie in what scientists call colonial integration, a form of neural coordination where signals pass directly from zooid to zooid through connected tissue, allowing the entire colony to respond as a unified organism, despite having no central command. It's a form of consciousness, if we can call it that is entirely distributed, entirely collective, and entirely alien to how we understand awareness.
Perhaps no discovery illustrates the otherworldly nature of siphonophores better than the recent documentation of their hunting postures. When the research team aboard the Falker encountered that massive Apolemia specimen in 2020, they weren't just documenting size, they were witnessing behavior. The spiral formation the siphonophore had adopted was unlike anything previously documented. Instead of the typical vertical curtain of tentacles, this colony had arranged itself in a massive horizontal spiral, creating what researchers described as a UFO-like feeding posture. The purpose became clear when you understand the three-dimensional nature of deep-sea hunting. By spreading its tentacles in a spiral plane, the siphonophore created a trap with no escape route. Prey approaching from any angle would encounter the web. The colony had essentially transformed itself into a rotating net, slowly spinning in the current, maximizing its catch potential in an environment where every calorie counts.
The ecological importance of siphonophores cannot be overstated. In many deep-sea ecosystems, they are among the most abundant predators, occurring in such numbers that they represent a fundamental link in the food web. The common siphonophore, Nanomia bijuga, is one of the principal predators of krill in many ocean regions. Studies in Monterey Bay revealed that, at times, the combined population of Nanomia in the bay consumes more krill than all the whales that come to feed in those same waters. These delicate, nearly invisible creatures are eating more than some of the largest animals on Earth. Their impact on deep-sea food webs is so significant that many species of squid and fish larvae have evolved elaborate camouflage specifically to mimic siphonophores, decorating themselves in patterns that make them look like toxic colonies to avoid predation by larger hunters.
But siphonophores are not invincible. Despite their deadly arsenal, they fall prey to specialized predators that have learned to navigate their defenses. Certain species of narcomucid jellyfish hunt siphonophores exclusively. Gastropods with thick shells can withstand nematocyst attacks long enough to consume the soft bodies of the zooids. Other siphonophores, immune to their own kind's venom, practice cannibalism. The ocean sunfish, with its thick, leathery skin, can bite through siphonophore colonies with impunity. And perhaps most bizarrely, tiny translucent crustaceans called phronema, sometimes known as pram bugs, have evolved to chew their way inside siphonophore bodies, hollowing them out and living within their gelatinous structure like macabre hijackers, often killing the colony in the process. The life cycle of a siphonophore begins with sex, one of the few moments when these colonial creatures acknowledge their origins as individual organisms. Specialized reproductive zooids called gonozooids produce either eggs or sperm, depending on the colony's sex. Some species are dioecious, meaning individual colonies are either male or female. Others are monoecious, producing both male and female gametes, but at different times to prevent self-fertilization. When conditions are right, gonozooids release their genetic material into the water column, and if fortune favors them, egg and sperm will meet in the darkness. From this union, a single fertilized egg begins to divide. But unlike in other animals, where cell division leads to a multicellular individual, in siphonophores, this first cellular mass will become the probud, a specialized structure whose sole purpose is to produce more zooids through budding. The probud undergoes fission, splitting repeatedly, each new bud developing into a different type of specialized zooid. Pneumatophore first, then nectophores, then gastrozooids, bracts, gonozooids, all emerging in a sequence programmed by genes that are identical in every cell, but express themselves differently in each developing zooid.
The colony grows, adding new specialized individuals in repeating patterns, lengthening by feet, by yards, eventually by hundreds of feet, all from that single fertilized egg that chose cooperation over competition. The evolutionary success of this strategy is undeniable. Siphonophores have existed in their current form for over 500 million years. They survived the Ordovician extinction that killed 85% of marine species. They persisted through the Permian extinction, the worst mass die-off in Earth's history. They witnessed the rise and fall of the dinosaurs, the advent of mammals, the emergence of humans, and the technological revolution that now allows us to finally observe them in their natural habitat. While countless other lineages went extinct, unable to adapt to changing conditions, siphonophores endured by never changing what worked.
Modern research into siphonophores has revealed details that border on the fantastic. Studies using high-speed cameras have captured the tentilla strike mechanism, showing conformational changes that occur faster than the human eye can perceive. Genetic analysis has traced the evolutionary relationships between different siphonophore species, revealing that the most ancient group, the cystinects, lack swimming bells entirely and simply drift with currents, while the more derived groups evolved the nectophore structures that allowed for active swimming. Researchers have discovered that the exact number and arrangement of zooid types is species-specific, like a biological barcode that identifies each kind of siphonophore. And perhaps most intriguingly, evidence suggests that the ancient ancestor of all siphonophores possessed more types of zooids than most modern species, meaning that siphonophore evolution hasn't been a story of adding complexity, but of streamlining, of losing unnecessary zooid types to create more efficient colonies.
Yet, for all we've learned, the siphonophore guards its secrets jealously. Because these creatures are so fragile, disintegrating at the slightest touch, live specimens are almost impossible to collect and study in laboratory settings. Everything we know comes from observations made by remotely operated vehicles and from the painstaking examination of fragments that survive collection. Questions that seem simple remain unanswered. How long does a siphonophore live? We don't know. How fast can they grow? The data is incomplete. How do they find mates in the vast emptiness of the deep ocean? The mechanisms remain mysterious. Do they have any form of memory? Any ability to learn from experience? The distributed nature of their nervous system makes this question almost impossible to test.
Standing now at the edge of understanding, looking down into the abyss where these creatures hunt in darkness, we're forced to confront the limits of our knowledge. The deep ocean covers more than 60% of our planet's surface, and we've explored less than 5% of it. Every expedition into the depths returns with new species, new behaviors, new mysteries. The giant apolemia discovered in 2020 may be the longest animal ever documented, but somewhere in the unexplored trenches and canyons, there may be siphonophores even larger, colonies stretching farther than we can imagine, drifting through realms of crushing pressure and absolute darkness that we may never reach.
The siphonophore challenges our understanding of what it means to be an individual, what it means to be alive. Is a colony of thousands of specialized beings working in perfect coordination one creature or many? Where does individuality end and the collective begin?
These aren't just abstract philosophical questions. They cut to the heart of how we define life itself. The siphonophore exists in a space between concepts, neither fully singular nor truly multiple, a living paradox that has been solving problems of existence since before our ancestors left the ocean.
As we retreat from the depths, ascending back toward the surface world of light and air, the siphonophore remains below, suspended in its element, tentacles spread in patient embrace, waiting for prey that might not arrive for days. It doesn't hurry, it doesn't despair. It simply continues its timeless drift through the darkness, as it has for 600 million years, as it may continue for 600 more. The deep ocean keeps its secrets well, and the siphonophore is one of its most closely guarded mysteries, a string of horror in the abyss, a colonial ghost that defies our attempts to categorize it, understand it, or even comprehend what we're truly looking at when we see it spiraling in the darkness. And somewhere in some unexplored canyon, deeper than any human has ever ventured, the longest siphonophore ever to exist is hunting right now. Its tentacles spread across hundreds or perhaps thousands of feet, a living net cast into the void, patient as death, older than mountains, alien as anything that swims in the space between stars. The ocean holds it gently in the crushing embrace of the deep, and it waits, eternal, hungry, impossible, real.